I'd like to welcome everybody this morning to the Four-State Dairy Nutrition and Management Conference webinar series. It continues today with Assistant Clinical Professor of Precision Dairy Nutrition at the Pennsylvania State University, Dr. Leoni Martins, who will be discussing recent research evaluation and soybean processing characteristics, laboratory methods used to assess heat-treated soybeans, and the application of processing on protein and fat nutrition in dairy cows. This presentation will complement presentations at the Four-State Dairy Nutrition and Management Conference next week. The Four-State Dairy Nutrition and Management Conference represents the latest research on nutrition and management of dairy cattle and has expanded the conference impact on the industry with the webinar series. This conference and webinars are a collaborative effort of the University of Illinois Extension, Iowa State University Extension, University of Minnesota Extension, and the University of Wisconsin Extension. I ask that you please click on the evaluation in the chat box at the conclusion of the program. It's short, and your input helps us to improve future programs. Dr. Martin's research and extension programs focus on precision nutrition and its interactions with lactational performance and nutrient use efficiency in dairy cows. His goal is to advance feeding strategies and optimize nutrient delivery to cows, enhance animal performance and health, and reduce the environmental impact of dairy production. As I always say, Dr. Martins, the podium is yours.
All right. Thanks, Fred. And thank you all for taking the time to be here today. And today I'm going to share some of the results we have from a work that we started probably a year and a half ago, looking into processing and feeding of soybeans in commercial dairy farms, mainly in Pennsylvania. And we know that this is a hot topic, especially now that we have these milk markets shifting a little bit with protein prices going up, butter fat prices going down, and we are probably looking into ways to increase milk protein synthesis in the cows. And what I like to say is that probably in the past 7 years with the high oleic soybeans, we have focused a lot on the fatty acid components of these soybeans, but not necessarily we've been looking into the protein fractions. So my work is kind of more tailored towards the protein fractions and how the processing on farms are affecting those. So quick overview of the importance of this topic. As I said, we're probably looking into strategies that would increase milk protein synthesis. And we know that increasing milk protein synthesis is a concerted action of hormones and nutrients, all right? Back in the day, probably we all have been taught that as a rule of thumb, when we're trying to increase protein yield, we probably should be pushing more starch in the diets or changing starch fermentability. And those strategies would probably increase milk output as well, not just protein yield. But those strategies are not necessarily friendly when we think about milk fat synthesis, all right? So if I'm increasing starch, well, I'm decreasing that rumen pH and probably putting that cow in a situation that she's more prone to milk fat depression. But things have changed a lot in the past few years. With genetic selections, we've seen these Holstein cows producing a lot of butter fat. And we should probably start thinking about these interactions of how the mammary gland take those nutrients and how the rumen functions. And we can actually increase both components or actually push more protein in these cows without really hurting butter fat. This is just a statistical analysis of some data that I put together for cows enrolled in different experiments at Penn State in the past 10, 15 years, just to draw those relationships. So how much of the changes in protein yield are actually explaining changes in milk yield? And we can see that there's a pretty strong relationship there with an R square of about 76%. So changes in protein yield explain about 76% of changes in milk yield in this data set. When we look at milk fat, well, the relationship is still positive, although that R square is way smaller, so about 45%, right? But the relationship is still positive. And one other thing that these statistical models can help us understand or make some inference on the biology of the cow is that in a hypothetical scenario where the cow is producing zero protein, she would be expected to produce 1.5 kilos of milk. All right. So those are very intricate relationships. So if the cow is not producing protein, she's probably also not producing lactose. And then she doesn't have that drive for milk synthesis. All right. On the other hand, if I suppress fat production in that cow, well, the statistical model still tells me that that cow would produce around 16.5 kilos. All right. So the relationship is not that strong, but again, it's positive. And the way that I look at this is that with the modern cows we have, the genetics we have, and the knowledge in nutrition that we developed in the past decades, we can certainly optimize rumen fermentation to actually make those cows produce more milk, more protein, and not have a very negative effect or a negative effect at all in milk fat. And high oleic soybeans, they come handy into this concept because it's a feed that is rich in protein. It's about 40% protein and also fatty acids. So if we include those soybeans in the diet and we have a well-formulated diet, most likely we can have some positive results. So the first data we have with high oleic soybeans was published back in 2017. This work was done at Penn State by Alex Ristov's group, and they looked into replacing extruded soybean meal with extruded Planish soybean meal or whole Planish soybean meal. And these soybeans, they were processed at 320 degrees Fahrenheit for the extrusion or roasted at 317 degrees Fahrenheit for the whole beans. And energy corrected milk didn't change when they made those modifications in the diet. And those were the inclusion levels that they fed to these cows, 17% for the extruded or 7.4% for the whole rolled high oleic soybeans. The response was in milk fat, and that makes sense because we are bringing that oleic acid into the diet, and that oleic acid will help with fatty acids digestibility and will help with the energy partitioning of the cows, and the response in butter fat was there. All right, so in terms of yield, there was a tendency going from 3.4 for the extruded soybeans versus 3.5 for the Planish soybeans. Moving forward, work out of Wisconsin, they evaluated the processing of these conventional and high oleic soybeans fed to dairy cows, so 20 hosting cows at 88 days in milk on average. And the soybeans were fed 16 to 18.6% on the diet dry matter intake. And one thing we need to note about this study is that these soybeans were all raw soybeans. So the authors didn't want to have a potential confounding effect for the roasting process here, and they did want to investigate the effect of processing. So what is going to happen if I feed ground or whole, either conventional or Planish soybeans to these cows? And it's interesting that in that particular study, feeding the Planish beans actually reduced milk production of the cows. So there was a decrease of about 3.6 pounds per day when feeding the Planish, either ground or the whole beans compared with conventional. But again, the milk fat response was there, and it makes sense. We're reducing that linoleic acid. We are putting the cow in a lower risk for milk fat depression, and the cows actually responded to that. Now, that response was only when I was grinding the soybeans. So if I increase the surface area of those soybeans in the rumen, well, the challenge for milk fat depression increases in the ground conventional, but not really, it doesn't really change for the ground planish. All right. So these were the milk fat yield responses, about 3.3 pounds with the ground conventional and 3.6 pounds for the ground planish beans. So couple or a few years later, work out of Michigan State, looking into the dose response of these high oleic soybeans in the diet. They fed these 24 hosting cows that were around 122 days in milk. Soybeans in those cases were ground at 771 microns, and they were roasted at 315 degrees Fahrenheit. And they were fed in the diets going from 0 to up to 24%. And the way that they worked with these diets was replacing soybean meal with and soy hulls with the high oleic soybeans to achieve those inclusion levels. All right. So all the dietary nutrients remain pretty much the same except for the fatty acid content. And their responses was pretty substantial going from 0 to 8 or 16 and 24. But there's a caveat for this because the 0% diet was the lowest diet in fatty acid content. So it's not, it's probably an effect of energy more so than an effect of high oleic soybeans. But nevertheless, these authors reported increased milk fat, linear effect, and true protein, both linear and quadratic fat, both content and yields. All right. And I guess a main takeaway of these studies that these cows can handle a lot of these high oleic soybeans in the diet and still keep the same energy corrected milk or milk yield. All right. So up to 24%, they kind of did just fine. So in a follow-up study, the same group, Adam Locke and his student, published this paper looking into the effect of roasting comparing with raw soybeans. And they picked kind of the middle ground from the previous study. So they fed 16% ground high oleic soybeans in a corn silage-based diet. Their treatment was a control diet with a lower fatty acid concentration, just as the previous study, and then the addition of the roasted beans or raw soybeans, which is the blue line, or raw soybeans plus a bypass protein source. In this case was amino plus. All right. So the main question here is if I'm feeding the raw soybeans, can I add that bypass protein from another soybean source and still get the same response? And I guess these results were quite interesting because when comparing roasted versus raw, there was a 7 pound difference in milk production, showing that, all right, we roast those beans, we protect that nitrogen from ruminal degradation, we increase the bypass protein, and the cows responded to that, right? But when looking at the raw versus raw amino plus, the response was still there, but it was way more modest, all right, just about 2 pounds. So this study was quite interesting to observe because that tells me that it's not just about roasting the soybeans and providing that bypass protein, but there might be something else in those soybeans that are fed raw that may compromise the efficiency of these cows in converting that amino acids into milk protein or milk synthesis. All right. And thinking through these data, one thing that comes to mind is the presence of anti-nutritional factors in soybeans. We know that soybeans have these anti-nutritional factors. Some of them are inhibited by the roasting process, which we call them the heat label anti-nutritional factors. Trypsin inhibitors would inhibit the trypsin action in the intestine and prevent or decrease that digestibility of amino acids in the intestine. We have some other anti-nutritional factors, but we do have reasons to believe that the responses that we observed in the Adam Locke study from 2024 might be related to these anti-nutritional factors. And unfortunately, we don't have good data looking into the effect of these anti-nutritional factors into the metabolism of the cow. As we learn more about gut integrity, inflammation, immunomodulation, probably it makes sense that the presence of these anti-nutritional factors would be integrated into those responses, and we don't quite see the same response as feeding roasted versus raw, just because there might be something at the intestinal level. All right. So when we think about soybean process, there are pretty much two ways to process those soybeans. One is temperature, and we showed some data on the effects of temperature, and the other one is particle size. All right. Uh, Recommendations for particle size and also temperature, they kind of date back from late 80s, early 90s. And when we search for information on how to properly process the soybeans, that's the data you're going to find. So roasting should be between 290 degrees to 315 degrees Fahrenheit. Uh, Following roasting, the soybeans should be steeped for 30 minutes. Often the temperature of those soybeans will be 10 to 20 degrees Fahrenheit cooler than the roasting temperature. Once these beans are steeped, we move them into a cooling bin. We get rid of all that heat, and then the soybeans are ready for storage. All right. These processes, when it's done in the industry, it can be quite controlled. The industry would have means to control each of these steps and take care of the quality of these processes. But there's a big question mark on how these processes would look like when we see farms doing more of these on-farm roasting processes. All right. And that's probably where the variation would come from, and that's probably the point of control that we need to take care moving forward. There's usually a price associated with that at least in the Northeast region, ranges from $35 to $45 per ton. And then particle size recommendations, as I said, a couple of studies back in the 90s would indicate that grinding these beans at halves and quarters or quarters and eighths would result in the best response in terms of digestibility and performance of the cows. These particle size would equate about 12 to 1400 microns, all right? And more recently, as you could see by the trials that I showed in the first slides, people have been targeting this 750 micron particle size, which there's not a whole lot of research backing that data, but the rationale behind using those values is that when you look at the particle size of bypass protein sources such as AminoPlus, that's the particle size that AminoPlus has. So by logic, you can understand that 750 micron would be adequate in terms of the protein fractions, right? But I kind of challenge a little bit this number because probably this will depend on the diet that we're feeding, on the cow that we're feeding, and how much of the soybeans we have in the diet. So when I was looking at these recommendations, I started asking myself, well, the cow has changed a lot in the past 40 years. We now have these high oleic soybeans being fed quite a lot in the diets. So would these recommendations still hold true for the current conditions we have, especially with the on-farm processing? All right. So what should be the roasting temperature? What should be the steeping duration? What kind of metrics should I use to evaluate whether I'm doing a good job with roasting on farm or not? And then would that depend on feeding levels and particle size? So with that in mind, we proposed this project to the Pennsylvania Soybean Board, where our objective was to characterize the particle size, nutritional value, usage strategies, and economic benefits of feeding roasted soybeans. And pretty much what I was looking for in this study was to learn what the farms are doing, since we don't really have good research papers to back up the current feeding strategies that we have with high oleic soybeans. So let's learn with the farmers, let's see what they're doing, and maybe we can use that data to generate some specific questions to follow up with controlled studies. So we visited 14 collaborator farms across Pennsylvania, and we actually received 5 samples from New York and Wisconsin that we included in our database. For the farms that we visited, we planned 2 visits with a 10-week interval just to capture some of that within farm variation. And on top of collecting soybean samples, we also performed a feeding assessment because the ultimate question would be, do farms that process their soybeans more appropriately would also have higher milk production, at least in an observational study. And here I'm going to show some of the results that we got. Preliminary data of this study was actually presented in last November during the Penn State Nutrition Conference. And now I'm presenting kind of the full data set. We still have some analysis to complete, but we have pretty much 90% of these project completed. So you will have firsthand access to this data. All the graphs that I'm going to show will follow kind of the same pattern. So these are violin plots. It's a nice way to see the distribution of the data across the y-axis. So each of the variables that we're looking for are described on top of the plot. In the orange or red color, we have the conventional soybeans, and in the green or blue, we have the high oleic soybeans. All right. Uh, the widest part of the plot, it represents the majority of the samples. So where the majority of the samples were within that analysis. All right. And right off the bat, when we were looking at these results, dry matter was quite variable. We had samples going from 85 to up to 96, 97% dry matter. All right. And that might have some implications on the roasting process or the quality of that roasting process, because many processors would add some moisture into the roasting procedure just to help the soybeans to cook more properly, right? So if we are cooking a soybean or roasting a soybean that is 85% dry matter versus 96% dry matter, we have reasons to believe that they would cook differently, all right? So probably the cooking process with a more moist soybean would be more uniform and better. The other thing that, or the other implication we can come with this is about storage, right? So we develop a good relationship with the soybean processors that visit farms and roast these soybeans on a daily basis. And some of them started reporting to me that it was common to see soybeans being roasted at less than 80% dry matter. So I'm wondering what is going to be the shelf life of this product when you put that in a feed bin and you keep it stored for a few weeks, right? So this kind of a serious conversation, because we are putting the soybeans in a challenge of developing some of these mycotoxins and spoilage, all right? Crude protein was not that variable, although you may think looking at the graph, but I would invite you to observe the y-axis. The range was from 38 to about 43%. Uh, this kind of aligns well with some data we have available from the University of Illinois. Uh, in that university, they collected soybeans across the US, and they different sources, different processing methods, and they fed those to swine. And there was very minimal variation in terms of the swine response, in terms of weight gain and also digestibility of amino acids in the intestine. So I would assume that this very similar to our case, that crude protein doesn't vary a lot. Ash is also variable, and that's probably related to contamination. Again, when you're processing the soybeans in industry, probably you have a better quality control on cleaning those soybeans, removing things that don't belong, and having a more uniform product, opposed to when you do that on farm, you're not really deholing those soybeans, cleaning them up. You're just hosting everything that is in the feed bin. Just make a point on the allowable storage time. We have some data from North Dakota State University for unroasted soybeans. And interaction between moisture content and grain temperature. Although we don't have this information for roasted soybeans, I would say that it's safe to apply some of these recommendations to the roasted soybeans as well. So the highest the moisture we have and the highest the temperature we have in the grain, the lowest the storage time should be, because then we would have problems with spoilage. Going into fiber components, we have ADF, NDF, and lignin, and let's just focus on NDF here for a minute. And there was quite a big variation there, all right? So samples going from around 7, 8% to up to 25%, right? And again, I think that's related on the purity of that soybean that is being roasted, depending on whether you're doing that on farm or with a roasting processor. Now, what I would ask to the nutritionists in the room is that, would you plan your diet formulation differently just because if you know that your soybeans have 25% NDF, would that affect the way that you adjust your non-fibrous carbohydrate sources in the diet, or how you estimate the rumen function of that cow and how that pH will behave when we have such different NDF concentrations? And that becomes probably more important as we have higher inclusion levels of soybeans. Another interesting data to look at is the fatty acid composition. So we have total fatty acids on a percent of dry matter basis. Quite variable too, or not really quite variable, because the range in the X is not that big, but samples going from 19 to up to 27%. Most of the samples will meet those book values of 22, 23%. One thing that I would like to challenge you again is that go into your ration formulation software and look at the feed library values for roasted soybeans. If I'm not mistaken, at NDS or AMTS, that book value would be 18%. So probably when you start analyzing these soybeans more often, you may want to adjust that number up, because most of these samples are around 23%. All right. Oleic acid on a concentration on a dry matter basis or relative to total fatty acids. And let's just focus on the total fatty acids for a minute. Again, big range for the oleic acid, all right? So samples going from 66, 67% up to 88%, all right? And that can be quite a big difference, especially as we try to get more precise in the fatty acid formulation, all right? So that was quite a surprise, because all these soybeans were Planish beans collected in different areas in Pennsylvania, some of them in New York or Wisconsin. And one thing that we need to keep in mind is that there's a pretty strong and inverse relationship between linoleic acid and oleic acid. So the highest the oleic acid concentration in the soybeans, the lowest is going to be the linoleic acid, right? So as we get these soybeans that are different in the fatty acid composition, have higher or lower oleic, we can safely assume that the linoleic acid would be proportionally higher or lower as well. And that has to do with the geographic location. So think about how we select corn silage hybrids. We have short season, mid season hybrids that we select depending on the region, and that will be responsive to the growing degree days that we have in those specific regions. It's pretty much the same thing for soybeans, although we don't have a lot of data on that, and recommendations or different varieties that would be adequate for higher growing degrees days or lower growing degrees days. But as you grow soybeans in the northern part of the state, or of the country, you can expect a lower concentration of oleic acid. So the farthest north we go, probably the lower concentration of oleic acid we have. The other component to this is that the maturity that we harvest these soybeans will affect the concentration of these acids, especially in regards of how stressed those plants were right before harvesting. So if we get late season soybeans, for example, that are exposed to a frost damage, we can expect that those soybeans will have lower oleic acid and higher linoleic acid. So these are all agronomic traits that, we need to learn more about those. And as the industry develops, probably in the near future or in the far future, I would say, we may have different varieties that are specific or region specific and growing degree days specific. One thing that I would caution you is that we have some other varieties coming into the market. They're not quite high in oleic acid. They will be lower in oleic acid. So although we may call these soybeans high oleic soybeans, pay attention to how much of oleic and linoleic acids these soybeans have, because if I'm feeding 6, 8 pounds of the plenish beans and then I replace those plenish beans with the different variety that is lower oleic acid, well, I may put my cow in a big challenge for milk fat depression just because now I have higher linoleic acid concentration. So what about processing? These are the roasting temperatures that we observed in our study. Quite a big range as well. Some samples at 230 feet, 235 degrees Fahrenheit, going up to 304. These two particular samples were soybeans roasted in an electric roaster. These other samples were either roasted with an open flame on farm or in a roasting processor, and there will be some variation on the type of equipment. All right, and looks like that these electric roasters, they will roast the soybeans in a lower temperature than the open flame. And roasting duration was pretty uniform across all farms. I would say that 1 to 2 minutes was the transit time for those soybeans. We got two samples that are kind of outliers here, but that's what the farmer reported to us. They said about 7 minutes, but majority of the samples 1 to 2 minutes, and then steeping duration. All right. So all these samples on the bottom, they steep the beans for 20 to 30 minutes. All these samples on top, I maxed them out into 360 minutes because these actually were uncontrolled steeping and cooling processes that we were not able to determine how long it took for these soybeans to steep. The reportings were we steep these beans from 3 hours and some farms up to 72 hours. So these beans steep for a long time. All right. And then for those that are not familiar with the on-farm roasting, 29 out of these 38 samples that we collected were roasted on farm. So a big population of samples that came from these on-farm roasting process. This is the main equipment that we observed in the field. This Rostomatic 400. The beans will go into the roaster. The roaster is an open flame and has a drum that will spin those samples for 1 to 2 minutes. So that flame temperature can be set at different temperatures. In this case here for this picture was about 460 degrees Fahrenheit. And then after 1 to 2 minutes, these soybeans come out, and that's where we take that temperature of the soybeans. And in this case, was about 280 degrees. After roasting, the farm may have an option to transfer the beans into the steeping and cooling bin, which is pretty much an unit with a lot of fans that will blow that air, and the beans will slowly come down to the bottom. And as the beans are coming down, they are cooling. All right. So this is not really the same as a steeping in industry where we would restrain the soybeans in a closed container for 20 minutes before actually blowing out that hot air. Here, both things are happening at the same time, right? And this is prone to the environment, right? If it's 50 degrees outside, well, maybe we're going to get a different product compared if it's minus 20 degrees, right? So these things we need to take into consideration. All right, so what I like to say is that for our population of samples, we either had controlled steeping and cooling or uncontrolled steeping and cooling, because some farms would not adopt this third step in the process. And for two main reasons. The first one is the service provider may not have the steeping beam because this is a big cost for them to purchase that equipment. Uh, and the other option that I heard about is that the farm didn't want to steep and cool these beans with the processor because that would have an associated cost of about $4 per ton, which I think is pretty low compared with the potential benefit we have of controlling that steeping and cooling process, which I'll show some data later. But we don't have good data to support processing recommendations for these mobile on-farm roasters. But these may be a very critical step in the processing of soybeans on farm. Just for reference, how an electric roaster works, here we can kind of isolate the environmental factor because this would require a closed facility that we can kind of control the outside temperature better. But the soybeans would go through this electric roaster, so no direct flame. And then they would be dumped into this steeping bean. They would stay here for 20 minutes, 30 minutes. You can set that. And then these beans, once they're steeped, they would be cooled down and then moved into a mill. In this case, a hammer mill. All right. What about particle size? So these are the geometric mean particle size data. This is the standard deviation, surface area, and number of particles per gram of soybeans. So let's focus on the mean particle size. Quite a huge variation here. Samples going from 450 microns to up to 3,500. All right. So these would be the samples that are just cracked in halves. These would be samples that are really pulverized, all right? Very small particle size. What is interesting to observe is that most of the samples, they were below 1800 microns, which would be pretty much equivalent to that recommendation of halves and quarters. Just some pictures to help you understand how these soybeans look like. So these are the halves and the 3500 micron soybeans. You can see that there's little variation here for these soybeans. And as we grind them, there's actually an increase in the variation. And that's the aspect of a soybean at 400 micron, 750, and the 450 micron. All right. All these samples on top, they were ground with a hammer mill. This sample here on the right corner, it was ground with a roller mill. And what is interesting to observe is that the variation of these particles was much smaller than the hammer mills. All right. So it appears that grinding these soybeans with a roller mill will provide a more consistent product. And that's probably related to number of hammers we have in that equipment, the equipment condition, RPMs, and all that kind of stuff. Just some plots to show you the distribution of particles across the sieves. You can see here that when we have the hammer mill, these samples are spread across all sieves. For the roller mill, we have a larger proportion of samples concentrated just in these two sieves, so less variation. And what is interesting to observe is that all these distributions are not really normal distributions. So what is the implication of having some soybeans like this with a larger proportion of smaller particles when it gets into the rumen? How are we challenging the rumen with these variation in the particle size? So things to think about, and probably design some research in the future to help us understand the implications of this in terms of rumen fermentation. Main reasons for people to be willing to grind these soybeans as fine as possible is that nobody likes to see these soybeans coming out in the feces, right? And that makes sense. We always understand that as a loss of digestibility. So I asked my students to handpick the soybeans they could find in the feces. This is a preliminary data because we still need to estimate fecal output for these cows. But this is the number of soybeans that they found. Majority of the farms had around 30 to 40 soybean particles in the feces, at least the ones that my students were able to identify. One or two farms that had quite a lot. And when you transform that into a proportion relative to the fresh fecal matter, most of the farms were around 2%, all right? But some farms had quite a lot. So again, this is preliminary data. We'll have to investigate this a little bit more. What is interesting to observe is that when you look at these soybeans closely, you can see all these little holes in the soybean particle. And to me, that means that bacteria had access to those, right? So, what is the extent of degradation that we had for protein in the soybeans? Well, we'll try to answer that or at least have some indication moving forward. But that increased porosity, a question I have is, does this indicate digestion? Probably yes. Now, how much of digestion we have, we need to quantify that. So what about protein metrics? So we roasted the beans, we have the feed ingredient that now we can incorporate in the diet, but how do I know whether I'm doing a good job or not? Well, traditionally, we've been looking into ADICP, NDICP, PDI, and urease as metrics for heat damage or soybean processing, right? And there was quite a big variation there for these metrics. NDICP growing from around 2% to up to 12%. PDIs, most of the samples would be within range that we know as the optimal range, 9 to 11. A couple samples that were way above of that range, right? What is interesting is that I was talking with CVAS folks, and they told me that they can clearly see when the soybeans are heated because in the process of determining NDICP, they recovered the residues, they filtered the residues from the NDF assay to run the nitrogen assay into those residues, and they can visually see those proteins that are clumped together. That doesn't happen in the ADICP method because they treat the samples with strong acids, and those acids would precipitate the protein. So when they mentioned that to me, it clicked on my mind that maybe NDICP would be a better metric than ADICP if we can visually see that in the lab. So that may be an indication of the heat treatment. And I'll show some data later that may support or may not support that. Another option we have the ROS multi-step protein evaluation. So it was a modification of these multi-step protein evaluation that we had from many years ago. That's from Cornell, Mike Van Amberg and his student Debbie Ross. It's an in vitro process that we submit those samples to a rumen fermentation digestion, and then we mix those samples with enzymes to kind of simulate the intestinal digestion as well. And when you send the samples to that assay, you're going to get soluble protein, an estimate for RDP, RUP, and intestinal digestibility for all those fractions. All right. So let's focus on the RUP here. What got me thinking about this data is that the range went from about 30 or 45% to up to 85% in many of these samples. All right. And a question I had was if these soybeans are 88, 85% RUP, doesn't really make sense that the intestinal digestibility would still be that high. All right. So on average, we got 68% RUP with 85% digestibility of RUP or 90% digestibility of crude protein. When we look at feed library values, so for example, if you go to the NASEN database, the average RUP and RDP in their feed library will be around 50%. All right, so if you take those fractions A, B, and C, you can model those and find those values. So 50% to 68% is a pretty big jump. So we got the samples, we did our own in situs, and we got those fractions A, B, effective degradation, effective degradability, and RUP. So let's focus on the RUP we got from these samples. Again, the range of RUP was still there, but now we have about 54% on average, which is 12, 14% units less than the ROS RUP. All right. So, what would be the factors that affect these protein metrics? So, we did the relationships between roasting temperature, roasting duration, and steeping duration, and all these metrics that you see in the y axis. And the roasting temperature doesn't really make sense when we look at ADICP, NDICP, or even some of the in situ fractions. We know that as we heat feeds, we should expect to see an increase in NDICP, and actually the roasting temperature relationship is negative. So that doesn't make biological sense at all, right? And that is the same for the other metrics. So ROS intestinal digestibility, as I increase the roasting temperature, I increase the digestibility. That doesn't make sense. Now, when we look at the steeping duration, well, now things start falling into place. So when the ICP was positively associated with steeping duration, so the longer the soybeans cook, the more NDICP I get. The lowest the digestibility of that protein, the lowest the degradation rate of that soybean the rumen, right? So that does make sense. So when I look at these data, because there's probably not enough time that the soybeans roast through that roaster, so it's just 1 to 2 minutes, probably steeping duration will be more determinant than roasting temperature when looking to this protein metrics. And then how much of these variation can be explained by farm or between farm variance or within farm variance? So farm variance is depicted here in blue. So the darker blue means that more of the variance is attributable to farm, all right? And on the opposite direction, very little of that variance is between farm variance, but a within farm variance. And you can see that all these variables that are highlighted in red, farms explain very little of that. So a lot of random error in this or things that we didn't actually measure in this study. But all the other metrics, they had some substantial between farm variation, which indicates repeatability, right? So what we learn when we look at these data is that farms are different, and probably the decisions that they make to process the soybeans will be very consistent from visit to visit if they keep the same process. All right. So just to summarize kind of all these data, we saw that in situ degradability fractions, over 72% of the variance could be attributed to farm. When we look at the ROS, RUP, it was around 63% of the variance attributed to farm, PDI 76, and the ICP around 70%. When we look at the ROS intestinal digestibility and total tract digestibility, very little of that variance can be explained by farm. All right. Now, we send the samples to the lab, but how do we know we are close to reality or close to truth? Assuming that our in situ method is a good method to assess protein degradation, we know that in situ has problems, but we need to establish something as the ground truth so we can compare these other lab metrics. We draw the relationships between in situ RUP and ADICP, NDICP, PDI, soluble protein, ROS RUP, and ROS intestinal digestibility of RUP. And surprisingly, there was absolutely no relationship of these metrics with RUP, with the exception of PDI. So PDI was significant, the relationship, as we increase RUP, we decrease PDI. But look at the R square. The marginal R square is the variance that is explained by PDI alone when we exclude the random effects. So 22%. I'm not really comfortable with that number. All right. So that's a very little explanation, although it's still significant. And there's a lot of noise in this data. All right. What about fraction A? So in situ fraction A, all these are the same variables related to the in situ fraction A. Again, PDI tended to be significant, but with an R square of 0.19. So again, very small R square, not comfortable with that. What about fraction B? Well, now NDICP is the one that has a tendency here, but look at the R square. Not good at all. All right. The only metric that was more promising is the NDICP when we relate that to degradation rate. So how much of that nitrogen or protein disappears in the rumen per hour. Uh, and NDICP was significantly associated with degradation rate, and that R square is about 44%. So from all these metrics that we evaluated, NDICP seems the one that is more consistent in evaluating degradation rate. Now, we know for a long time that PDIs have limitations, so that has been acknowledged by the authors that put out this paper when they developed this protein dispersibility index. It's a good metric to answer a yes or no question, all right? So we can tell whether a soybean was not processed or very undercooked. But as we apply more heat or steeping into these beans, we can no longer tell the differences between those. All right. So PDI would tell you a yes, no question, but not really differentiate those soybeans or those different temperatures. And the ROS assay, well, we've been using these to measure or to estimate RUP, but the metric or the assay was developed to actually measure undigestible nitrogen or undigestible crude protein. All right. To calculate RDP and RUP, we would have different time points, just as we do in situ. So we could estimate the slowly degraded nitrogen pool, the rapidly degraded nitrogen pool, and then apply this equation. And that's different of assuming a 16-hour% retention time and assuming that whatever got degraded in 16 hours would be RDP, and whatever passed that 16-hour threshold would be RUP. The other limitation of these metrics is that none of them, with the exception of the in situ assay, would capture differences in particle size. And that makes sense. So for all these plots, we have the geometric mean particle size in the x-axis and all these protein variables in the y-axis. And many of these assays would standardize the particle size to 1 millimeter. So for example, NDICP, ADICP, and PDI. The ROS assay would receive those samples as is and analyze them with the original particle size, but they don't really relate to the geometric mean particle size. And we know for a fact that the higher the particle size we have in the rumen, the lower degradation in the rumen we'll have. Therefore, for RUP, we're going to have higher RUP, all right? And in situ is the only one that was able to capture that with an R square of 0.68. Now, how do these variables would relate to milk production, on-farm milk production? So remember, this is an observational study, so we're not trying to draw causation here, but more on association, trying to raise some questions that we may be able to design some experiments later on to investigate this. we had very few variables that tended to be associated with differences in milk yield. Some of them doesn't really make sense. As we increase rumen undegradable protein intestinal digestibility, we actually decrease or tended to decrease milk yield. Some of them do make sense. So as I increase the effective degradation of that nitrogen in the rumen, I decrease milk yield. We know that if I'm not providing that bypass protein, uh, probably that cow, high producing cow, would be short in amino acids. And on the other hand, as I increase RUP, I increase milk production, right? The other way to look at this, I mentioned that steeping is probably a big factor here for soybean processing. So let's separate those farms into those that control the steeping process or the ones that don't control the steeping process. So these were overcooked soybeans, let's put this way. How did the farms perform? Well, milk fat was increased for the controlled steeping process. Energy-corrected milk tended to increase for the controlled steeping process versus uncontrolled. Now, one point that we need to make is that these data may be confounded by the dietary composition, all right? Although there was no statistics here for crude protein or NDF and starch, well, these are pretty big numerical differences. When we fix the model or correct the model for the potential confounding effect, the statistical significance disappears, but we still have a 4 pound or 4 kilos energy corrected milk production difference. So again, we're not trying to draw causation here, but that's probably something that deserves some further investigation. Common questions that we get in the field is how much to feed. I've seen 1 to up to 12 pounds. Uh, Probably most of the people I talk would describe 6 pounds as the optimum. Common question we get is, can I feed it raw? We showed some data that you can, but probably we don't really understand the negative implications of those anti-nutritional factors. And how much of a premium should I pay if I'm feeding these soybeans? We're going to discuss these two topics, but really quickly, I would like just to make a point that depending on the software that you're using to formulate your diet, you may not be able to predict differences in milk production when feeding raw versus roasted, just looking into that nutrient prediction from the software. So for the nascent users, this is a diet that was exactly the same. I just replaced raw soybeans with roasted soybeans, and these are the predictions for milk production. And energy allowable, MP allowable, nutrient allowable, difference in true protein or fat, pretty much no difference. So the model doesn't really grasp those differences in the fractions A, B, and C. For the CNCPS users, well, that model is doing a better job. When I do the same diets or plug the same diet into CNCPS, the model actually predicts a 5% decrease in milk production. So if I am adjusting for that lower MP because I'm using raw soybeans, probably I would be able to correct for that difference. But again, we don't really know what's going to be the negative implication of anti-nutritional factors present in that raw soybeans. And then what about premiums, and would feeding soybeans pay off? This was an analysis put together by Chuck Nicholson at the time he was at Wisconsin. They collected some research data and made some assumptions on how much of these soybeans would be fed to the diet and what kind of milk fat response they would get. And they looked into milk prices and commodity prices in a 10-year time span from January 14 to September 24. And in all those situations, feeding the soybeans at 5% the diet dry matter would result in a positive milk income over feed cost. So it would be very beneficial to add soybeans into the diet. Now, this is very dependent on the milk fat response, all right? In the current market, I use their same model to plug in the numbers. if we're feeding the same amounts and we're expecting the same responses, that would be equivalent to still making a positive income over feed cost of 0.4 or 0.9 dollars per cow. So much modest or very small responses, but still we're not losing money. All right. Trying to expand that modeling work, I put together this sensitivity analysis depending on how much butterfat is paid. So going from 1.5 to 2.75 dollars per pound of butterfat, what would be the milk response going from 30 grams to 70 grams, and how much of these high oleic beans we would feed in the diet. So let's just get this example or this extreme example of butterfat being paid at $1.5 per pound, and that we're feeding 4 kilos of soybeans in the diet. Well, in this situation, we would be losing money, right? So about 10 cents less income or feed cost if we're feeding that level of high oleic soybeans, right? Now, if the response of butterfat is 70 grams, well, then, or 60 grams, then we start to see a positive relationship. So this is going to be just for your reference as you try to figure out, depending on the premiums you're paying, what kind of response, basically purely on milk fat response we would get with these high oleic soybeans. So just to wrap up, I think we learned with this study that there's substantial variability in the roasted soybean samples. One thing that I would encourage nutritionists is to start analyzing these soybeans, and it needs to be farm specific. All right. For most of these variables, we saw that between farm variation explains a lot. So it means that it's what the farms decide to do. You probably will get those responses in the lab analysis. Particle size we still need to develop in this area. The 750 micron may be adequate for some diets with moderate inclusion of soybeans, but if you're feeding higher inclusion levels or if your cows have a bigger challenge in milk fat depression, probably that 750 will be too small. We don't really have enough evidence to validate these metrics that are available for us to estimate RUP. So that's probably something we need to work on in research, is trying to create a model that would predict that RUP based on the current metrics we have. What I would recommend is use one of these metrics. So for example, NDICP, to track changes over time for specific farms. That's probably going to get you closer to the value that you need to have in your formulation software. And the best thing to do is to keep track of the cows, how they're responding. Are they milking as you're expecting? The MUNs are in the right place. The milk protein synthesis is good. So ask the cows, and they're probably the best ones to tell you whether this is being done properly or not. So with that, I would just like to acknowledge the collaboration with the Pennsylvania Soybean Board that provided most of the funds for this, United Soybeans, and also the collaborator farms and all these folks that helped us to put together this experiment. And I'll be glad to take any questions.
Thank you, Dr. Martin. We have a fairly small group. I would ask that folks just go ahead and unmute their microphone and ask the questions directly. I'm not seeing anyone unmute. I've got a question here. If the NASEM does probably the least accurate evaluation model for nutritionists, is there a consensus to create a change or a patch or just use a different model?
Yeah, so I don't want to get the impression that Nason doesn't do a good job in predicting things. I would just say that for specifically when we're looking into these protein fractions of feed ingredients, there's no model inside Nason that would track those changes. So maybe moving forward, that's something to actually incorporate into the system. The main problem with that is that many people don't measure those. And my understanding is that Nason tries to keep things kind of more simplistic for metrics that we really measure on farm, and then we can use to predict changes in milk response. But for example, Nason does a good job with predicting amino acid flow for some specific amino acids. So, for example, lysine, Nason does a better job than CNCPS in predicting the outflow of lysine to the intestine, right? And that's an advantage of the model. But when you're looking purely into the energy predicted milk production, or MP predicted milk production, I wouldn't look at those values in Nason, because CNCPS probably does a better job accounting for these differences in all the protein degradation fractions and so on.
Very good. Any questions from the audience? Jim, I've never done a program where you didn't have at least one question.
I got
So
a
what is the
question.
future of high oleic beans? Where do you think they fit in from an economic standpoint and from, I mean, it just seems like they're the hottest topic here in the Midwest right now. And I know you guys have more experience out in New York and Pennsylvania with them. So tell me the 3, 4, 5 year experience that farmers out there are having.
Well, I think that soybeans will be a nice tool to have in your diet formulation program or in your cropping strategies, because energy dietary energy may become way more expensive than we can expect. So, for example, bringing into the diet those calcium salts or those fatty acids, there are some projections that those things will become much more expensive. So, if you're able to have a homegrown fatty acid source on your farm, that's a nice asset to have. So, in terms of reducing feed costs, I think the soybeans could fit well. The problem is that we can't just think about the fatty acid portion of it if we're not doing a great job on the protein fractions, because that soybean still bringing 40% crude protein into the diet. So I think it becomes easier to justify the soybeans with high butter fat prices, and high fatty acid supplements prices. But as these things change with the market, now we need to be a little bit more cautious considering on how much it's going to take for me to roast these soybeans to have appropriate quality control on those, and then incorporate those in the diet. So I think they still have a space, uh, but they're not a new vitamin. They're not going to solve, magically change the income or feed costs in the diet. They will probably bring some cost savings as the market changes.
We had another question being asked there. Go ahead and ask that question.
Okay, thank you, Dr. Martins, for the presentation. I have been feeding some of these high leg soybeans, just learning how to feed them here in the Midwest. But I am struggling with a little butter fat on this particular herd. But I noticed that my starch level seems to be a little on the higher side. So as we go up on beans, I'm feeding about 6.5 pounds of beans today. Do I need to pull my starch down to help that butter fat? Is there a correlation there as far as energy goes with that?
So, what is the particle size of those soybeans?
We're between 450 to 600 microns.
So, two things, right? One is you adjust that starch, you bring in more non-forage fiber sources, and you kind of create the environment for those soybeans to work. Or you could probably increase that particle size and kind of reduce the challenge from the fatty acid side of things in the rumen, right? So we need to remember that the high oleic soybeans, they are not no risk. There is still risk for milk fat depression. They are just lower risk, all right? And especially when we don't really know what is the oleic acid and linoleic acid concentration in those beans, we may put the cows in a risk for milk fat depression. feeding at 6, 7 pounds of beans and grinding them that fine, probably that's what's going to happen, Either adjust the starch, but then we know that you could probably lose some milk there, and we don't want to see that. But one of the main things you could play with initially is changing that particle size. That doesn't mean you're probably going to start seeing more of those beans coming out in the feces. But again, my perception is that I wouldn't be concerned with those beans coming out in the feces, especially because they seem to be a very small proportion of the fecal matter. And we help the rumen with that larger particle size. The other thing we need to think about is that as we go lower in particle size, I'm also losing that RUP, all right? So there's a protein component to that. So you could go either way. Think about decreasing starch, bringing more non-forage fiber sources, or playing with the particle size a little bit.
Very good.
Right?
Go ahead.
Yeah, Nelson Laws here. Yeah, maybe I agree with you, Leoni. Maybe he's feeding like, I mean, that particle size is like baby powder. So as you say, it's not I mean, the oleic acid is not inert in the rumen, and it may be, what he may be seeing is some effect on the fiber digestibility. He's affecting probably the microbes, so that's why he's losing some of that milk fat. Anyway, so the question that I had for you, Leoni, was because of the difference between the spread of the particle size between hammer mill and roller mill. Roller mill is going to give you a more precise release of the oil and the protein in the rumen and all that. Do you see any value on the spread of the particle size that hammer mill delivers?
Right. That's a great question. And when I look at these data, from a theoretical standpoint, I have no doubt that having that big spread would definitely make things differently in the rumen, right? So those small particles would be readily available and probably compromise microbial fermentation, and you have that other subpopulation of samples that are larger particles that would take longer to digest. So I do think that that spread is a problem. Now, how much of a problem that is, we don't have good data on that for these high oleic soybeans or soybeans in general. But I do think that if I have the opportunity of using the roller mill to be more consistent in the particle size, I know that the particles that are going into the rumen, they are more consistent, and then I can predict changes better than if I have different populations of particles within the same sample. So that's an important observation, and I would agree 100% with that.
We're to the end of our time slot. Thank you, Dr. Martins, for sharing this information and your insights on feeding high oleic soybeans. I remind everyone that this program has been recorded and will be available on the Fourth State Extension Dairy Group website. Give me 4 to 7 days and we'll have it up. In addition, I will email you when it is available and include the link to the program evaluation. Please take 2 minutes and answer the questions in the evaluation to help us improve this webinar series. Thank you for your participation, and I look forward to seeing you at the webinar next month. I will close this webinar. Thank you for attending. Thank you, Dr. Martins.
Well, thank you. I hope that was useful. So.
Fantastic.